Everyone, today's webinar on a pioneering approach to treating autosomal recessive deafness 9, also known as ARD9, a serious genetic condition that leads to severe bilateral hearing loss from birth. We're honored to have Dr. Yilai Shu with us, a leader in the field of gene therapy for hearing loss. I'm Tiffany Penn from Packaging Marketing Team, and I'll be your moderator today. Dr. Shu will discuss the groundbreaking trial of AAV1 human OTOF gene therapy, sharing key insights from its development to its promising outcomes. This novel treatment has shown significant potential in improving the lives of individuals affected by ARD9, making a major advancement in the treatment of genetic hearing loss. This session will include a detailed presentation by Dr. Shu, followed by an interactive Q&A session, where you will have the opportunity to ask questions and deepen your understanding of the impact of this gene therapy. Simply put your questions at the Ask a Question box, and Professor Shu will help answer them towards the end of this webinar. We are delighted to have facilitated this exchange of knowledge and ideas in our quest to better understand and combat genetic hearing loss. And let's look forward to an engaging and informative session with Dr. Shu. Dr. Shu, all yours.
So today my talk is AAV1 gene therapy for children with FMB9. So I'm from ENT Hospital, Fudan University. So I will introduce a little bit background. And about 5% of population suffer from disabling hearing loss, and in China, there's about 70 million people suffer disabling hearing loss, and there are about 30,000 newborn deaf children per year. So the hearing loss affects speech, language, and cognitive development, especially in infants and children, and education and employment in adults, and for social isolation, the mental health for seniors. So there are about 26 million people suffer from congenital deafness, and 60% of are caused from genetic effect. And so far, about more than 150 genetic deafness genes already found. So so far, all of deafness genes, they are monogenic, and we can do local injection. So that's the advantages for inner-ear gene therapy. And the gene therapy method include gene replacement, gene editing, and RNAi and ASO. So the first target gene is OTOF mutation. It's called DFMB9. DFMB9 is a congenital or prelingual severe to complete recessive deafness. So it influence 2 to 8% of patients suffer from genetic hearing loss. So the mechanism we use is the gene replacement to do the gene therapy. So the OTOF gene is the size is about 6 kb. So it expresses single AV. So we use the dual AV mediated gene therapy to restore hearing in OTF knockout mice. So this the papers we published. We use the nucleic acid level and the protein level. Both of them can restore the hearing. So, and the second part is the preclinical studies. So the preclinical studies include drug components, including the drug delivery system, promoters, screening, and recombination strategy. And the second part is AAV vector production, and the optimization, manufacturing, precision, and finally this GMP production, and the safety and efficacy in mice, and the safety in monkeys. So the first one, we optimized the inner ear delivery routes, including the neonatal mice, adult mice, and the guinea pig and the monkeys, and we found the different routes, and they can target different specific cell types, and to do clinic the clinical trial, we also developed a new device. That's the device is specific for the human in-ear delivery. So the dual AAV strategy is here, including the AK and AP and the TS. From the Western blot, we found that the TS and the AK works well, and in the in vivo data, we found that the AP, we found that the AK is the highest efficiency. So we choose the AK and AAV1 combination to translate. So these are the preclinical studies for safety data. We studied the hearing blood test, and the histopathology and the etiology, transduction, and the biodistribution, and we found it's safe. And then we applied the clinical trial. So the first part, we we did the unilateral gene therapy for the patients with DFNBNI. So that's the drugs, the GMP drugs, actually it's produced by the pageant gene, and that's the ASIC approved, and that's the recruitment, and the patient's age is from 1 to 18 years old. The general principle is from older to younger children, because for the safety safety concern, the general principle is from older to younger children, and the low dose to high dose. And actually, the first recruit is 2022 October. So as you can see here, totally 425 patients with hearing loss, and 23 DFNBNI patients, and finally we choose the 6 patients for the first study. And the first first 3 patients, they are all older than 3 years old. The first one is 4.8 years old, and the second one is 5 years old, and the third one is 6.2 years old. And we wait 6 weeks, at least 6 weeks, to observe the DRT, if it's safe, and then we can move forward. And the fourth patient is about 2 years old. So here is the our injection route. So we inject through our external canal, auditory canal, from here. canal, and then that's the injection needle, and that's the endoscopic is and that's the cochlear, and that here is me. So after injection, no DRTs, dose limiting toxicity and serious AE, and we can see the increased neutralizing antibodies to AAV1, and the no T cells response to AAV. See, the first patient is a no dose. So the total 6 patients, they are all complete deaf, because we want to use the complete deaf. It's easier to detect it's effective or not. So so the baseline is from higher than 95 dB. 95 dB we call it complete deaf. It's like higher than 95 decibel, and the patients cannot hear. And the best, and after the injection, we can have the hearing restored to 68 dB at 4 weeks, and 53 dB at 3 months, and 45 dB at 6 months. And that's the ABR and the ASR. They are objective assessment for hearing restoration. That the subjective, the PTA. You can see here, that's the baseline, and that's baseline, and after treatment, we can see the hearing restored, and actually the hearing gradually getting better. And then we moved to the higher dose, and 5 patients, and four of 5 patients restored the hearing, and you can see here. And the best one is the patient 4. Patient 4 is from baseline to higher than 95 dB to 38 dB at 6 months. And then we can, as we can see here, the hearing actually gradually getting better. It's a time-dependent manner so far. So far, the longest follow-up is more than 1 year, and the hearing it's very stable. So we also observed the improvement of speech perception in 5 patients. So as we can see here, and here are 2 videos showing the before injection, before treatment, and after treatment. And before treatment, and the kids didn't have any response to sound, and after treatment, and they can respond to sound, and then gradually, and they can speech. Okay. So for the privacy protection, we mute the name and cover the eyes. 4 weeks, 10 weeks, he can look back. He can look back. And he can speak 6 months after injection.
重新说，我。
So he's older than 2 years old, and he's already 1 year old. 1 year after the injection, he can speak much better. And the second video showed the patient 6 and at baseline, and at different time points after injection.
Hello,
叫妈妈，妈妈。
小鸡怎么叫啊？小鸡怎么叫？叫妈妈，叫爸爸，叫奶奶，爷爷，苹果，香蕉，鸡蛋。
Okay, the patient six actually he can speak a little bit earlier than patient one, the patient four. I think because the treatment time is younger than patient four, and he's 1 year old when he had the treatment. Okay, so the conclusion is this is the first in human clinical trial with gene therapy for genetic hearing loss, and this first in human clinical trial of dual AAV gene therapy. No DRT and serious AEs occurred in patients after treatment, and 5 of 6 children showed hearing restoration, and with 40 to 57 dB reduction in average ABRs ratio. And hearing was restored in time-dependent manner so far, and we need to follow up longer time. And speech perception was improved in 5 of 6 children. And after unilateral treatment, and we carried out the bilateral gene therapy for patients because the bilateral hearing had better the sound resource localization, and in in the noise background, and the bilateral hearing had a much better detection, the sound detection. So almost the same. We followed the same principle. I mean, from older patients than younger patients. And the first patient is actually is 10 years old. No dose limiting toxicity and serious AE, and the increased neutralizing antibodies to AAV1 and no T cells response to AAV1. And the patient one is 10 years old. The right ear and both right ear and the left ear is from baseline higher than 95 dB to 58 dB at half year. And you can see here, as you can see here, right ear and left ear, and both the objective and subjective assessment. And the patient two. Patient 2, patient 3, patient 4, and patient 5, all of them had hearing restoration. So we also detected the ability of sound source localization restoration in all 5 patients, and the speech perception improvement in all 5 patients. So here, the first video showed the patient 1 before injection, and she didn't have any response to sound. After treatment. Different frequency.
Ba. I. U. S. Mama.
That's the sound localization test, and she can recognize where the sound resource, the sound source localization.
奶牛，对，马，
Where's the horse?
对，鸭子，
And she can understand which
对，
one is dark. She can understand. She not only can hear and she can understand. And then we can see the second.
Yeah. Yeah. Yeah.
No response to music before treatment.
Yeah.
After treatment.
Yeah. Yeah. Yeah.
叫爸爸，
叫奶奶、爷爷。
Okay, well, clearly we can see the hearing and the speech perception and the sound source localization restoration improvements. So to summarize, this is the first in human bilateral gene therapy for genetic hearing loss, and no DRT and serious AEs occurred in patients after treatment. And all 5 patients had a hearing restoration and speech perception improvements, and the sound source localization was restored in all patients. So finally, I would like to thanks for the platform of ENT Hospital for the University in Shanghai, China, and Professor Hua Li and Wu Qing Wang and Dr. Bin Chen's collaboration, and our team, the team from team include the postdoc doctors and students, PhD and MD students. And also I would like to thanks to the collaboration from Mass Engineer, Harvard Medical School, Zheng Yichen and Ren Chai from Southeastern University. And especially I would like to thank Refresh Gene Therapeutics and Dr. Rachel Qingqiu Tan and Liu Xinxin, Kai Gao, and the funding. And finally, I would like to thank PackGene to produce the GMP AV production for us. Thank you.
Thank you, Dr. Shu, for this enlightening presentation. And for our participants, feel free to put in your questions in the Ask a Question box below the broadcasting image, and we will receive them and then answer them as we receive. We already have a couple of questions in the list, so we're going to go through them, but feel free to put in more, and Dr. Shu will answer them as many as the time allows us to. So let me start with the first one. Professor Shu, what is the long-term safety monitoring measures in place for the participants?
Sorry, say it again. Sorry.
So what is the long-term safety monitoring, for those who participated this trial?
So we from our plan we were operate to have 5 years then follow up for the safety and efficacy.
Okay, so like how long would that process be? Are we talking about like maybe a couple years or
Oh,
even
right now? Right now, it's the longest one is 1 year, 1 year and 4 months already.
but
And
you will continue to monitor that participant?
yes. So 5 years totally. Yeah, that's our plan.
Okay, great. So, afterwards, if there are any other discoveries throughout this 5 year span, please share with us. Let's move on to the second question. So, how does the hearing recovery relate to different dosage of the therapy?
Um, that's a good question because we consider the OTF is the real disease. So the lower dose and low dose and high dose, um, we can see the high frequency, the higher dose had a little bit better hearing compared the cell termini. We can see the uh, different frequency, the high frequency and the hearing can restored a little bit better.
I see.
Mm.
Okay, let's move on to the next question. How does this gene therapy compare with other hearing loss treatments?
Yeah, it's quite good question. So for treatment, the present treatment for congenital deafness, um, the only way is hearing aids or cochlear implants. The cochlear implant, the CI cochlear implants.
Mm-hmm.
There's no any available clinic drugs for them. So compare I think the gene therapy is um, the etiology therapy. It's a drug, and the cochlear implant actually they can restore the hearing to some degree, but it's it's not natural. It's not natural sound. It's not
Mm,
natural. That's the biggest thing. And the other the other thing, this cochlear implant actually is artificial uh, implantation. So that's actually the disadvantage include in the background noise, the detection, the hearing detection is not that good. It's not that good, and for the music detection is not that good. So we are comparing the new treatment gene therapy to CI right now.
I see.
Mm.
Okay, thank you. So the next question we have right here is actually a two-part question. So the first one is, how soon after the virus injection did the children first show a response?
Um, 4 months, about 4 weeks.
4 weeks for the
Yeah,
first shown after the injection.
yeah, because the 4 weeks is the first time we detect um, the hearing test. Actually, for some of them, they can have a sound response between two to 3 weeks.
Okay, so the second part of this question, did any children show signs of discomfort because they were not used to perceiving sounds?
Hmm. Sorry.
So did any of the children who received the treatment show any signs of discomfort because they were not used to receiving sounds?
Oh, yeah. So they actually because the actually four the total is 11 children, 11 children, and four of 11 they already had 1 year cochlear implants, so they can detect the hearing. They can detect sound before, but if the CI and the cochlear implants turned off and they cannot hear anymore. And the rest of them, say seven children, they hadn't they they hadn't hear before, and after treatment they can hear and they can turn back. They can turn back, and if you call him behind him, and he can turn back.
Okay, thank you. And then the next one we have here is would a gene therapy address different OTOF gene mutations?
I didn't understand it.
I think it's just as asking if like other sort of gene mutations can be treated with this
Oh,
AAV
you
treatment.
mean other deafness gene?
Yes.
Other deafness gene is most likely successful. You mean, right?
Yes.
Okay, so the OTOF is the first gene had the successful clinical trial so far, and other gene already we actually we already had almost 20 genetic deafness genes successful in animal model.
Mm.
Animal model, but in the human clinical trial, the autophony is the first one. So we are working hard to translate some of the like a KCNQ4, TMC1, and GJB2, and the MyC6. They are all work in animal model.
Okay, so I guess that leads to the next question we have here. So are you going to try to make treatment for gene CJB2?
Yes, that's because GJB2 is the most common deafness gene. So it's very important. It's about 2.4 million people.
Okay, thank you. So moving on to the next one we have here, is it possible to do bilateral treatment but subsequently? Is treatment of the second year planned for this first patient cohort?
Yeah, that's a good question. The first batch of patients actually we did the 1 year injection, and then we are considering to do the other year. The other year, we are doing the animal study, and so the neutralize the antibody. It's effective for the results or not. So far we actually we from our animal data we found that if we do 1 year and then this subsequently do another year, the same works. So I think it's okay because it's local injection. We use very small small drugs.
Okay, thank you so much. And the next one we have is there a potential explanation for the non-responder patients?
Sorry?
Is there a potential explanation for the non-responder patients?
Oh yeah. The patient two. The patient two, actually the 11 patients, only one patient didn't have respiration. Probably reason is the neutralized antibody, because the baseline we found that the patient 2 had the highest neutralized antibody, because AAV is almost is everywhere else. Somebody already have the same AAV, and he can have the neutralized antibody. That is the possible reason. That's a possible reason.
Okay, thank you. And then I guess we have a follow-up question from the previous one. So when can we expect your research on the CJB2 treatment?
I don't know. We are working on the animal model. The GGB2 actually is harder than autofolding, but the GGB2, the GGB2, they have very big population. It's really worth to do.
Okay, so I guess once we have some results, we'll definitely share with the audience. And moving on to the next one. So did the treated children show issues with their balance?
Nope.
Okay.
Okay.
And then the follow-up question, does the vector enter cells of the vestibula?
Yeah, from the animal model, we can see the virus can affect the utric. Yeah, the utric is like a vestibular system. Even they can affect autohealth cells. But when we use the autofolding antibody, they don't show the autofolding positive immunostaining. So that means somehow the cochlea can control the expression of the autofolding.
I see. Okay. I think that's all the questions we've received during the presentation. If you do have any other questions, feel free to email us at PacGene, and then we'll help relay those questions to Dr. Shu, and then he can help answer them if you still have questions. Thank you very much for your engagement and throughout this webinar. And if we do encourage the attendees to continue to explore the possibilities of gene therapies. And we look forward to seeing you at our future webinars. Thank you once again for joining us today for your time. And thank you very much, Dr. Shu, again for today's presentation. And that concludes today's webinar. Thank you.
Thank you, Ms. Peng. Thank you, everyone.
Thank you. Bye-bye. Have a good day.
You too.